molecules. If I have correct information, you already had neurotransmitters with Dr. Tenka. Yes, so today we will continue with the signaling molecules that typically do not behave as neurotransmitters. That means the steroid signaling molecules. Then we will repeat the nitric oxide that you know from the previous course. Then thyroid hormones, and then the peptide hormones. So if we start with the steroid signaling molecules, we begin with the fact that you know from the previous course. You know that when we spoke about the synthesis of cholesterol, we mentioned that cholesterol is precursor for some signaling molecules that we call steroid hormones. And we also defined that we have 5 groups of steroid hormones that are listed here. That means gestagens, representative is progesterone, androgens, that means, for example, testosterone, estrogens, that mean estradiol, glucocorticoids are represented by cortisol, and mineralocorticoids are represented by aldosterone. And after this lecture, you should be able to define the steps how to synthesize these steroid hormones. If you will look at the synthesis, there are many of intermediates of the synthesis, but you do not have to know all of them by name, only those that I will show to you. Yes? So that means that absolutely crucial for you are these 5 steroid signaling molecules, for each group 1 member. If we start from the very beginning, the synthesis is quite simple. We take the substrate. The substrate for all of them is cholesterol, and you will see that the first phase is the same for all steroid hormones. And then in different tissues, you will have different enzymes that will take the intermediates of the synthesis. The location inside the body, you typically produce steroid hormones in some specific tissues, in some specific cells. I think that with histologists, you describe these cells producing steroid hormones. So how can you identify them? So, tubular mitochondria, then lipid droplet, and yes, smooth endoplasmic reticulum. So that's the typical triad for these cells. And therefore, it's not surprising for you that the steroid hormones will be produced in mitochondria, the beginning and end phase, and the majority in the smooth endoplasmic reticulum. Yes, that's the location. Inside the body, these hormones are produced typically in suprarenal gland, in the cortex of the suprarenal gland, where you produce androgens, where you produce mineralocorticoids and glucocorticoids, and then in ovaries and testes, where you produce the sex hormones. That means typical androgen in testes, as is testosterone, and estradiol and progesterone, typical female sex hormones in ovaries. Then, what we will mention at the very end, we have also some tissues that can to some extent convert the steroid hormones each other. For example, in adipose tissue, you have the aromatase that's able to do the peripheral aromatization of the androgens. But the whole pathway is not typically located there. So 3 main organs means testes, ovaries, and the suprarenal gland. Where do we produce them? As you know, for steroid hormones, we cannot speak about the storage. Since steroid hormones, as you know from the signaling part of this course, can have the intracellular receptors, they can cross the membranes, and therefore you cannot store them inside some vesicle in the producing tissue, in the producing cells. And therefore, steroid hormones are produced immediately after the signal. On the other hand, if you want to transfer them inside the plasma, you must have some transport protein. Since they are hydrophobic, they cannot be transported freely in the plasma, but they have some specific or unspecific transport protein. Unspecific one is, for example, albumin. Specific one is, for example, sex hormone binding globulin or cortisol binding globulin. More about them you will hear later this year when you will speak about the plasma proteins. Yes, but at least these 3 you should know from today.
Can you repeat the names?
sex hormone binding globulin and cortisol binding globulin, where if you have somewhere BG means binding globulin, and the letter between means what does it bind. So you, for example, have vitamin D binding globulin, or you have here cortisol binding globulin. Or you can have thyroid hormone binding globulin. Here you have that they have the cytosolic receptors, but that I think you already know, since you are quite far in this course. So you know that the steroid hormones have the receptors not typically at the membrane, but they have typically them inside the cell. And you also know what's their mechanism of action. That means that they modulate the transcription of the target cell. If we start with the synthesis, so we know that the source is cholesterol. Question is, what is the source of the cholesterol? And as you know, you have two options. First option is to take the cholesterol that were produced somewhere else. That means to take the cholesterol from the LDL particles. That means that the cells producing steroid hormones will expose the LDL receptors, catching the LDL particle for the blood circulation. And this is the predominant way how to do it. Some amount of cholesterol can be produced de novo inside the cells, but that's the minor portion. That's not the main portion. It means that the first thing is the internalization of LDL particles, the cleavage of that particle, and release of the cholesterol from the particle. Next step is, since the first reaction is located inside the mitochondria, you must concentrate the cholesterol to the mitochondria. And this is done by the protein that we call steroidogenic acute regulatory protein, abbreviated as STAR protein. And here is written that is ACTH dependent. ACTH means adrenocorticotropic hormone. Yes, that's one hormone that regulates the whole synthesis of steroid hormones, and therefore it's also mentioned here. But if you write only STAR protein, steroidogenic acute regulatory protein, as the transporter of the free cholesterol, it's fully correct.
Do we need to add the entire names in the test, or the abbreviation is enough?
I think that this is better, but if you write only this, it's also good.
Good.
If you do not, if you, I cannot tell you this since I do not know what's inside the short answer part. Or I know it since I prepared it, but I will not tell you. But it's not enough to put there transport protein. So you must mention at least the STAR protein.
Okay.
So that's the first two steps. You must gain the cholesterol, and you must put it into the mitochondria. And then in the mitochondria, you have first irreversible reaction that is the same for all steroid hormones. There's the reaction where you do the side chain cleavage of cholesterol. All of you must know how many carbon atoms has cholesterol. So how many? 27. If you take 27 minus 6, it will be 21. And therefore, in this first step, you will convert 27 carbon atom molecule of cholesterol to 21 carbon atom steroid that we will call pregnenolone. You do the cleavage of the side chain. So if you look at the picture, this is the cholesterol. Here, I think this is from Harper, so they didn't board with the hydrogens, but it doesn't matter right now. And as you see here, you do the side chain cleavage. That means cleavage of the side chain of the cholesterol. And you remove these 6 carbon atoms. As you see, they are released here. And here you gain the molecule of pregnenolone that has 21 carbon atoms. This enzyme has the short name, that means side chain cleavage enzymes. That means SCCE, or you can call it as the whole cytochrome P450 side chain cleavage enzyme. About cytochrome P450 isoenzymes, I think you have already heard a little. You will hear about them much more when you will speak about the detoxification of xenobiotics. That's a very big group of enzymes and isoenzymes that do quite stereotypic reactions. And one of them is this sidechain cleavage enzyme. Yes? And since it needs for its activity the cytochrome P450, therefore it has it inside the name. So here for the test it's enough if you write sidechain cleavage enzyme. Yes? If you write it together with cytochrome P450, it's even better. This is the place where we have the pregnenolone. The pregnenolone itself is not active, and it must be subsequently converted to other steroid hormones. And here you have some of them listed. So if we make a simplification, we can say that we have 3 groups of steroid hormones depending on the number of carbon atoms inside the molecule. Progesterone, cortisol, and aldosterone, that's missing here. That means gestagens, glucocorticoids, and mineralocorticoids have 21 carbon atoms. Androgens have 19 carbon atoms, and therefore, as you see, you remove this residue of the side chain coming to testosterone. And then if you look to estradiol, that's a representative of estrogens, altogether the estrogens are missing this one methyl group that's here, and therefore estrogens have only 18 carbon atoms. And therefore it's not surprising that all of them can be produced from pregnenolone. Only for different group of the hormones you will use different enzymes.
Only 70 methyl estradiol has an aromatic.
Not all estrogens.
All estrogens.
That's per definition. But all of them have also aromatic ring and 18 carbon atoms. Yes? So if you have the scheme and you will say, good, but I do not know what is this, but you ask me on the group. So if you count the number of carbon atoms and it's 18 and we ask you on the group, you should write estrogen. Yes? The same if you read there 19, you can write androgens. And you do not have to know what precisely it is. Yes? So this rule is quite useful. What enzymes we will use to convert pregnenolone to those steroid hormones? We will use several types of enzymes. So for example, we will use hydroxylases that will use the molecular oxygen and NADPH. Then we will use dehydrogenases, and we will use the lyases. Lyases typically reduce the number of carbon atoms in the molecule. Yes? And what do we expect you to know is for the key steroid hormones to know what is the series of the steps. So this is the first simplified version that is useful for you to know. We can say that the pregnenolone is the same for all, and then pregnenolone is directly converted to gestagens, to the progesterone. And then progesterone and pregnenolone, they serve as the splitting point for the synthesis of 3 groups of steroid hormones. For glucocorticoids, for mineralocorticoids, and then from androgens. And then androgens works as the precursors for estrogens. So you cannot produce estrogens from mineralocorticoids nor from glucocorticoids, but you will produce them from pregnenolone, from androgens, and then you gain the estrogens. This is also for you useful if you will gain an information that some enzyme is missing. That means that you have inborn error of metabolism, and you know what enzyme do you use for synthesis of what. Then you will know that, for example, if you have blockade here, you still can produce androgens and estrogens. Yes, and there is one clinically significant disease where you have a block of one enzyme here, and therefore the androgens can be still produced. Since they have the common precursor, therefore if here is no defect and the defect is here, then you can still produce androgens and estrogens. And also here we have the number of carbon atoms that are inside the molecule. So if we start with the C21 group, first progesterone. Progesterone is produced directly from pregnenolone. Yes, the reaction I will show you in a few minutes. So that's the first direct single-step conversion. And then from cortisol and aldosterone, you have, we can say, 3 steps. And some steps are the same and some are unique. If we start with cortisol. Cortisol is produced from progesterone, and there must be the sequence of the hydroxylation in this order, as this is not a mistake, that the 11 should be the first, since the 11 hydroxylation is the last, yes? That means that the order should be first hydroxylation on the 17th carbon atom, then hydroxylation on the 21 carbon atom, and hydroxylation on the 11th carbon atom. So simply, if you see 21 carbon steroid with 3 hydroxy group on these carbon atoms, you can be pretty sure that it's cortisol. Aldosterone, as you see, shares some hydroxylations, but the first hydroxylation is missing. That means that aldosterone is also produced from progesterone, but then you have the hydroxylation on 21 carbon atom, on 11th carbon atom, and what's unique for it is the enzyme aldosterone synthase that produces the aldehyde group on the 18th carbon atom. So if you see aldehyde group on the 18th carbon atom, you can be sure that it's aldosterone. And if we look at the reactions, so here you have the cholesterol. The red means that it's in mitochondria, and that's the Sidechain cleavage enzyme. So here we have the pregnenolone with 21 carbon atoms. When it's green, it means that it's done in the smooth endoplasmic reticulum, the reactions. So as you see, one-step conversion from pregnenolone to progesterone, and the only thing what do you modify is this hydroxy group and this double bond, as that is the isomerized. The enzyme is called 3-beta-hydroxysteroid dehydrogenase, and it's the enzyme that converts or interconverts, since it's a reversible reaction, pregnenolone and progesterone. But typically you do it in this order. Then if you want to produce cortisol, the cortisol is produced by 17 hydroxylase, 21 hydroxylase, and 11 hydroxylase. Yes, so enough is to know 17, 21, 11. And then you can identify the cortisol that has 1, 2, 3 hydroxy groups and 21 carbon atoms. Second molecule that you want to produce is aldosterone. And aldosterone is also produced from progesterone. But you do not do the 17 hydroxylation, but you do the 21 hydroxylation, then 11 hydroxylation, and then here, by aldosterone synthase, produce the aldehyde group. So if you see the aldehyde on the C18, you know that it's the aldosterone molecule. These intermediates that you have written here, if you will learn next year or the further years, you will learn that some of them have some activities. And for some of them, you can identify that they have glucocorticoid activity or that they have the mineralocorticoid activity. And when you will learn the enzyme defects, you will learn that some enzyme defects will be different from the other. For example, if you have defect here or if you have defect here, the result will be different since you will produce different intermediates. But you do not have to take this into account right now. Only learn the enzymes that lead to the production of these 2 main representatives. It means cortisol and aldosterone. Question?
For example, cortisol,
Yes.
can be cortisol as well?
Yes, this you can do, but you typically do not do it. Yes, you know, that's the fact is that different, if you look at the in the cortex of the suprarenal gland, the cells in different layers have different enzymatic equipment. That means, and the same is in ovaries, so that means that not all cells have the full enzymatic equipment. So zona mineralosa produces mineralocorticoids, and zona fasciculata produces the glucocorticoids. And they have different enzymatic equipment. Any question? Then androgens and estrogens. Androgens have C19, that means 19 carbon atoms. If you want to produce androgens from 21 carbon atom steroid, you must remove 2 carbon atoms. And as you know from the second course, if you do some lytic reactions, some cleavage, you typically use lyases. And therefore, this shortage by 2 carbon atoms is done by enzyme that's called 1720 lyase, or abbreviated simple lyase, you can say. So that's the first part of the synthesis of androgens that you will, from progesterone or pregnenolone, start, and you will remove 2 carbon atoms. And this is irreversible step. You never can return. What is done is that when you do this cleavage, you will produce the keto group. Yes, if you chop this side chain by the lyase with pregnenolone or progesterone, here you will produce the keto group. And if you want to produce the full active and the main male androgen, that means testosterone, you must reduce this keto group to hydroxy group. And this is done by 17-beta hydroxysteroid dehydrogenase. Simply, if you say the reduction, the dehydrogenase, it's enough. Yes? So it's enough for you only to say, I reduce keto group to hydroxy group. That means that if you see 19 carbon atom steroid, that has here keto group, and here has the hydroxy group, it's testosterone. Here we have 2 intermediates, that's androstenedione and dehydroepiandrosterone. These molecules also have some androgenic activity, but they are not the androgens that are typically produced in big quantities in testes. Yes? This is the product of the suprarenal gland. But of course, these androgens can be essential, for example, for women. Yes? Since their ovaries do not produce testosterone, they produce directly the estradiol. So that's the testosterone, so 2-step synthesis. The first is -2 carbon atoms by 1720 lyase, and then a reduction of keto group to hydroxy group, and 19 carbon atoms. If we look at the last group, that means the estrogens. The estrogens have 18 carbon atoms, and the enzymes that's producing them is quite unique enzyme in human metabolism, and it's called aromatase. As you see, the aromatase does at one moment quite many things, but what we can say is that here you lose the methyl group, you produce here the aromatic ring, and here you have the hydroxy group. And if you see this we call the core for estrogens. And it's the typical sign for the estrogens. If we return here, then here you see that only estrogens has the aromatic ring inside. So if you see the molecule of steroid hormone with aromatic ring, you know that it must be estrogen. You count the number of carbon atoms, and it's 18, so you know to what group does it belong. In this full scheme, you see 2 other estrogens. One molecule is called estrone, and the second molecule is called estriol. Does anybody of you know what's the difference between these 3 estrogens? That means estradiol, estrone, and estriol. That's one thing. And the other. Aromatic ring, they have the same, all of them. The difference is the number of hydroxy groups. Yes, that's the first. Estradiol, 2, 1 and 2. Estriol, 1, 2, and 3. And estron has only 1. That's one difference. That's the chemical difference. And why do you think that we have 3? Typically, in different periods of women's life, you have different estrogen. Estradiol is the main estrogen since the puberty until the menopause. Estron is the main estrogen after the menopause, since it's produced by the peripheral aromatization. That means aromatization of suprarenal androgens. You see that it's not produced from testosterone, but it's produced from androstenedione. And then estriol, it's produced with the help of the baby and the placenta of the fetus and the placenta, and therefore estriol is a typical estrogen of pregnancy, of pregnant women. So 3 estrogens and 3 periods of the life. Women, estriol after the puberty, before the menopause, then estron after the menopause, and estriol during the pregnancy. More about them you will hear the next year in the last course, in the whole year, when you will speak about the endocrine system. Yes, but these 3 names you should have somewhere in the head, and they have the different role. Last molecule that's here is this, that's dihydrotestosterone. And here we have enzyme called 5-alpha reductase. So what do you know? Have you heard about dihydrotestosterone? So what is it?
It's responsible for the testes formation.
The dihydrotestosterone is something what we can say the fully active androgen. Testosterone has some intrinsic activity, but dihydrotestosterone is even stronger. And some peripheral tissues express the enzyme called 5-alpha reductase, converting testosterone to dihydrotestosterone, and we can say that these tissues are even more sensitive to androgens. Yes, so you, for example, have this in prostatic gland, yes, or in hair follicles. And dihydrotestosterone is more active, intermediate, than was testosterone. So from some point of view, we can say that testosterone is precursor for more active androgen, and also precursor for the estradiol, that means for estrogens. But also it has its own enzymatic activity, and you will measure it in the blood. But in some tissues, it's converted to other compounds. How is possible then in women's body we do not produce, or do you think that in women's body do we produce testosterone? Yes. And how it's possible that in ovaries we do not produce it and do not release it to the blood? Yes. If you learn about the ovaries, you know that there are different layers of the follicle, and the outer layer will convert the testosterone to estradiol. So it's true that in women's body you have testosterone, but to a big extent it's converted to estradiol. And the same, in male body you have estrogens, and they are typically produced by the peripheral aromatization of androgens in the adipose tissue. And if you have highly obese males, they have the estrogens also, since they have higher amount of adipose tissue. So saying that androgens are only male sex hormones and estrogens only female sex hormones is simplification, since you can measure and they can be produced both in both sexes. Only each group is predominant in one sex. So question to this. Regulation. Regulatory steps are the first 3. Internalization of LDL, star protein, and sidechain cleavage enzyme. That are the 3 regulatory steps of the whole synthesis. And the regulatory signals are the hormones that signal to the cell that the hormone should be produced. That means it can be the pituitary hormones, for example, adrenocorticotropic hormone, luteinizing hormone, and follicle-stimulating hormone. Yes, adrenocorticotropic hormone sends the signal for cortisol. Luteinizing hormone sends it for progesterone and for testosterone, and follicle-stimulating hormone sends it for estrogens. And then angiotensin. Angiotensin is the molecule that sends the signal for the aldosterone production. But angiotensin is not produced in pituitary gland. It has some complex metabolism, and it is part of the axis called renin-angiotensin-aldosterone. Yes, and about this axis you will learn later. About the degradation, several simple rules. First rule is we cannot cleave steroid core in our body. That means that we must modify the molecule, but we cannot cleave the core. So typically what we do with the steroid hormone is that we modify the molecule by some hydroxylations, and then we will conjugate it with something. And this something can be, for example, glucuronic acid, or it can be sulfates. And then these metabolites are excreted in urine, and in urine you can measure these metabolites. Second option is, since some small part of the unchanged molecule is excreted also, you can measure this unchanged part. And then we call it, for example, urine-free cortisol. Yes, but that's only the minor fraction. The majority are the metabolites that are produced inside the liver from the steroid hormones. If you have a liver failure, if you have liver disease, second, that is here mentioned only for you do not mix it with steroid hormone, is vitamin D and calcitriol. Question is this steroid hormone? And why not?
It is for
It can be produced in the body. Therefore, the term vitamin is not fully correct here.
It's a derivative of vitamin D.
It's true, but it's steroid hormone.
It's also derivative of cholesterol.
Yes, it's derivative of cholesterol. Since in our body we can produce it, the vitamin D we have the food store, or we can produce it in the body. Yes, it's produced from the cholesterol, from the intermediate of cholesterol synthesis that's called 7-dehydrocholesterol. But why it's not a steroid hormone? Since in the first step you do the photolytic cleavage and you cleave the steroid core, one ring in the steroid core. And therefore, in the final molecule that's called calcitriol, you do not have the steroid core. And therefore, it's not steroid hormone. So the thing is, it's derivative of the precursor of cholesterol, of the 7-dehydrocholesterol, but it's not a steroid hormone. But with steroid hormones, it shares some characteristics. Yes, typically, it also acts inside the cells. Yes, but per definition, it's not steroid hormone. You use the skin for the first phase, and then liver and kidneys for the 2 hydroxylations. Yes? That means that the full molecule of calcitriol, it has its name, in its name has 3 hydroxy group, calcitriol. And no steroid core, therefore no steroid hormone, but also produced from the precursor of cholesterol molecule. So it's very similar, but it's not the steroid hormone itself. And quick repetition of the nitric oxide. Nitric oxide you know from the previous course, nothing new. Nitric oxide is signaling molecule. It's produced from arginine, that's the essential amino acid. And it's produced by the enzyme nitric oxide synthase or NO synthase. And you convert arginine to citrulline, and here you release the nitric oxide. And you know that it has 3 tissues where it has very high activity: in the neurons, in endothelial cells, and in our immune system. Yes, and in each of these tissues, the function is slightly different. Maybe about the function in CNS, you will speak this year, or you can wait till the next year when we will speak about it on 100%. So this is also a molecule that you can find inside the test. Yes, it's in signaling molecule, and you should be able to say how it's produced and how it's degraded. That's quite simple. It's the free radical, so the half-life is very, very short. Any question? So if no question, then we will take the break till 15. So, second big topic for today is the formation of thyroid hormones. With Docent Renka, I think you spoke about catecholamines. Is it true?
Maybe.
Maybe.
Joking, yes.
Yes. And this is second big group of signaling molecules, the thyroid hormones, that are produced from amino acid tyrosine. But in the shape of the molecule, you can see that there is a very big difference. And the difference, if we say 2 main things, is that here you have iodine, and second, that here you have 2 aromatic rings. That means that for synthesis of 1 thyroid hormone, you need 2 molecules of tyrosine, you need iodine, and third important thing is still here you have the carboxy group. And therefore it's not a biogenic amine. And the whole synthesis is absolutely different from the synthesis of catecholamines. The only thing what's similar or was the same is that it's produced from tyrosine molecule.
What is the last group which we need?
That here you still have the carboxy group, therefore this is not a biogenic amine. Catecholamines are amines. Thyroid hormones are 2 main thyroid hormones. Triiodothyronine is 1, abbreviated as T3, that means 3 iodines, and thyroxine T4 means 4 iodines. And I can tell to you that the thyroxine is the precursor for triiodothyronine, since in some peripheral tissues you will cleave this iodine off and you will produce the fully potent thyroid hormone that's triiodothyronine. But thyroxine itself has also some intrinsic activity, so it's very similar story to testosterone and dihydrotestosterone. Both are signaling molecules. Only dihydrotestosterone and triiodothyronine are typically produced in the periphery, in the tissues that express some specific enzyme. But before we come to the periphery, we must first synthesize the hormones. So here you have some specific characteristics for thyroid hormones. First is that this signaling molecule must have a trace element. If you look at catecholamines, you need tyrosine. If you look at GABA, you need glutamate. Yeah? So typically amino acids. But here you must have the organic molecule plus the iodine, that means the trace element. And this brings the first problem, since there are some regions on the Earth where you have lack of iodine in the soil. And that means that these regions have, or could have, typically before 100, 200 years, and later, they had the problems that the people there could have deficiency of the thyroid hormones. Nowadays, if you know that there is some region with this risk, you do the fortification of iodine to the food. So for example, here in Czech Republic, since we were also one of that region, we have the fortification of salt. Yes? So the salt here is typically iodinated. That means that in all pastry or in simple salt, you have the iodine. And therefore, nowadays here we do not have deficiency. But some regions have this. The basic rule is the more far you are from sea, the bigger problem for the iodine deficiency in potential. That means the central regions of the continents typically could have these problems, and here we are in Central Europe. Second big difference from other signaling molecules is that if you learn about, for example, glutamate, catecholamines, everything happens from some simple molecule, yes? But here, you do the synthesis inside a very big protein molecule that we call thyroglobulin. So we can say that the first step of the synthesis will be the production of very big precursor molecule that's rich in tyrosine residues that we call thyroglobulin. And the changes will be done inside this one big precursor molecule. Third big difference is that typically for neurotransmitters, you learn that neurotransmitters are produced inside the cell. There you have the GABA shunt, there you have the synthesis of acetylcholine. Here, very big part of the synthesis takes place in the extracellular space. And if you look at the histology picture of the thyroid gland, we call that pink lake colloid. And that means that the reactions are located in the colloid, in the extracellular space, on the molecule of thyroglobulin. And only the beginning and the end phase you have inside the cell. But many reactions you do outside the cell. And then the last that I already mentioned, that there is the peripheral conversion, yes? But that's not unique only for thyroid hormones. So if we start with the first three steps. The first three steps simply corresponds with the fact what we mentioned on the last slide. So first is that you must produce the big precursor protein, thyroglobulin. It's protein, therefore you must produce in endoplasmic reticulum and Golgi apparatus, and subsequently secrete it into the colloid. Second thing is that you must synthesize the enzyme, and also you must put them into the extracellular reservoir, where you can do the reaction. And third is that you must take the traced element from the blood and put it to the thyroid gland. And this third step is done by the secondary active transport of iodine. It's sodium iodine Symporter, that's the molecule that does it, abbreviated as NIS. And here you have everything. So that means you take sodium, you take iodine, and Symporter means that both compounds, or here both ions, are transported in the same direction here inside the cell. So sodium moves along the concentration gradient, and it takes the iodine together with it inside the cell. You are able to express the efficiency of this transporter by something that we call TS ratio. TS ratio means what's the difference of concentration of iodine inside the thyroid gland, inside the thyroid cells, and inside the serum, therefore TS. And if you have a normal diet, the ratio is around 25 to 1. That means 25 higher concentration of iodine in thyroid gland than in serum. If you have iodine deficiency in the food, therefore the person has it also, the transporter will work even more actively and the ratio will grow, yes, since it tries to catch as many iodines as it can. This transporter is under a regulation of the superordinary or signaling molecule, that's TSH, that means thyroid gland stimulating hormone or thyroid stimulating hormone, and it's produced from the pituitary gland. That means the same as was ACTH, LH, or FSH for steroid hormones. Also here the thyroid gland has the superordinary gland, the pituitary gland. So is this clear to you? What do we have right now is precursor molecule. Thyroglobulin, we have the iodine, and we have the enzymes. And what right now we want to do is to put the iodine to the thyroglobulin molecule. To do this, we have the enzyme that's called Thyroid Peroxidase, abbreviated as TPO. Why you should know this abbreviation? Since you will check for the antibodies against this enzyme in the screening of thyroid gland disorders. Yes, so you will look at it, or you will read about it in the list of the things that you can measure in the blood of patients in the hospital, the TPO. What's the function of this enzyme is quite simple. It's a thyroid peroxidase, that means it uses hydrogen peroxide to modify the iodine and to help to incorporate it into the thyroglobulin molecule. How it's done? It's exposed to the luminal surface, that means to the colloid, and it converts the iodide, that means iodine -1, to the atomic iodine. There's the first possibility, that's the radical, or to iodinium, that means to some plus charge. So simply you can say that it's the oxidation of iodide, that means I-, to some positive charge. And these forms can subsequently react with the tyrosine inside the molecule of thyroglobulin. Thyroid peroxidase, it's called due to the fact that it needs to have hydrogen peroxide for the oxidation step. Thyroid peroxidase is also the enzyme that you should know. So you should know the transporter and this enzyme, since if you will meet the people that have hyperfunction of thyroid gland, you will use inhibitors of this enzyme, as for example carbimazole or propylthiouracil or uracil, to block this enzyme and therefore to reduce the function of the thyroid gland. And since thyroid gland disorders are the most common endocrine disorders that you will meet, therefore you must know how is called the regulatory enzyme of the synthesis. If we look at a picture, what do you do? So here you have the thyroglobulin molecule, and this is tyrosine residue. And if you do the iodination, you will put 1 or 2 iodines on the molecule. According to this, you will speak about monoiodotyrosine or diiodotyrosine. And there's the abbreviation MIT or DIT. The T means tyrosine, since it's done on the molecule of amino acid tyrosine, and you have here 1 or 2. Therefore, monoiodo or diiodo. But the whole thing is still done inside the big thyroglobulin molecule, so that there are no free tyrosine residues in the colloid. And then in the next step, that we call coupling, you do a very simple thing. You combine these two molecules together. And it's not surprising that if you want to produce thyroxine, that means T4, you will combine 2 molecules of the iodotyrosine. Since here you want to have 4, therefore you must 2 times 2. If you want to produce triiodothyronine, that means T3, you combine 1 molecule of monoiodotyrosine and 1 molecule of diiodotyrosine, and you have triiodothyronine. This coupling reaction is done also by thyroid peroxidase. So you can say that thyroid peroxidase does the oxidation, the production of the positive iodine, plus it does subsequently the conjugation. That means the production of the actually active thyroid hormones. But the coupling reaction still is inside the molecule of thyroglobulin. So you only remove the aromatic ring and you couple them together. Is this clear to you? That you simply, if you look here, that you see that the coupling is not done by peptide bond. Yes, that you directly transfer here this aromatic ring to it, and here you have, this is the full tyrosine, and here you take only the aromatic ring. Yes, so you do not do the full coupling. Yes, only one as whole and part of the other. So that's written here, and in the test can be questioned what is precursor for what thyroid hormone. All these steps are done inside the colloid. So after this phase, we can say simple thing. Everything is in colloid, nothing is active. The thyroid hormones are inside the molecule of thyroglobulin. Therefore, we have to return inside the cell, and in the last phase, you will release the thyroid hormones. How do you do it? By very simple process of the endocytosis. So the thyroid cells will take part of the thyroid. They will produce the endosome. The endosome will fuse with lysosome, and in lysosome you will degrade the thyroglobulin molecule. And subsequently, by the proteolysis, you will release the active thyroid hormones, that means T3 and T4, and you release them into the bloodstream. So is this clear? Of course, that in this phase, you do not release only T3 and T4. The problem is that still in the molecule of thyroglobulin, you have some molecules of uniodinated tyrosine and the molecules of monoiodotyrosine and diiodotyrosine. And the question is how you can recycle these molecules. And the recycling is done by one enzyme that we call central deiodinase, or the full name is iodotyrosine deiodinase. That means that it does the deiodination of the molecules of iodotyrosine. And it doesn't matter whether it's monoiodotyrosine or diiodotyrosine. Yes, therefore, iodotyrosine. And this enzyme takes the molecules of monoiodotyrosine and diiodotyrosine, removes the iodine from it. The iodine you transfer to the colloid, and the tyrosine molecule comes back to the endoplasmic reticulum and is incorporated into the new molecule of thyroglobulin. Why we cannot do it directly? Since monoiodotyrosine or diiodotyrosine, they have no transport RNA. That means they cannot be directly incorporated into protein. You know that into protein we can put only 20 or 21 proteinogenic amino acids. And monoiodotyrosine nor diiodotyrosine are on the list, so they cannot be incorporated. If we now return to the story of the thyroid hormones. So T3 and T4 are released into the bloodstream from the thyroid gland. What do you do in the blood is very similar to steroid hormones. you must bind them to some transfer protein. You have the non-selective one, that's albumin, and then you have the selective transport protein. That's abbreviated as TBG, that means thyroxine binding globulin. The abbreviation BG is the same, binding globulin. T means what does it bind, so thyroxine binding globulin. These molecules transfer the thyroid hormones through the body, and then in the peripheral tissues, they can do the signaling. You know that they have also intracellular receptors, only as the difference from steroid hormones, these receptors for thyroid hormones are typically inside the nucleus. So they are nuclear type of intracellular receptors. And if we return to the story, whether T3 or T4, in the periphery, you can find an enzyme that you can call as peripheral deiodinase, or you can say that it's iodotyronin deiodinase. Iodotyronin means that it's the thyroid hormone itself. It's not only one tyrosine. Yes, this was iodotyrosin, and this is iodotyronin. And this enzyme does selective one reaction when it takes one iodine from T4, it removes it and converts the molecule to T3, to triiodotyronin. And this is more active thyroid hormone, and it can show more activities in the peripheral tissue that's able to produce it. But the thyroxine itself has also the activity, but we can call it as the prohormone for more active T3 molecule. So any question to this? No question? So if no question, then the last topic for today are the peptide hormones. You know, the peptide hormones or peptide signaling molecules are a very big group of compounds. Here you have only some of them in the list. So peptide signaling molecules you can find in your central nervous system. For example, opioids, as is, for example, enkephalins, endorphins, are peptides and produced in the CNS. The molecules that we mentioned today, as is, for example, TSH, ACTH, luteinizing hormone, or follicle stimulating hormone, are also peptides. That means the pituitary hormones are typically the peptide hormones, and they send the signal to the peripheral endocrine glands what they should do. The same pituitary gland has the supraordinary gland, the hypothalamus, and the compounds that are produced hypothalamus are typically also peptides. Main hormones of the intermediary metabolism, of the energy metabolism, insulin and glucagon, are also peptides. Cortisol is not, but insulin, glucagon, growth hormone, that are peptides. Then many of growth factors, as erythropoietin that induces the hematopoiesis, the production of red blood cells, colony stimulating factors, or insulin-like growth factor are also typically the peptides. And very big group of intestinal hormones, as is, for example, gastrin, or as is secretin, are typically also peptides. So that means that this group has very big amount of members, and of course we do not expect you to know all of them. What do we expect you to know is that you will know the general steps how to produce the peptide signaling molecules, and here you have two real examples how it's done. Yes, but today we do not expect you that you will know the amount of amino acids of all of them, the sequence of amino acids. Of course, you never have to learn this. Yes, unless you really want to do very good science or very specific science inside. So what do we expect you to know is simple the steps from the synthesis, from the beginning of synthesis till the production of the final peptide signaling molecule. So the first, if you want to produce protein, what you must do? You must start in the nucleus and you must do the transcription process. Then you must go to ribosomes and you must do the translation process. And what you will produce is the molecule of the pre-pro protein. If we use the term hormone, we can call it pre-pro hormone. And if we take the insulin molecule, we can say pre-pro insulin. This molecule is typically transported into the endoplasmic reticulum. And the transport into the endoplasmic reticulum is enabled by the signaling sequence, and we can say that that's this pre. Once the protein is transported to the endoplasmic reticulum, you cleave off the signaling sequence, and you now have the pre-propeptide. That means pre-prohormone. Sorry, you have only propeptide. That means prohormone. And we can say proinsulin. So splitting off the signaling sequence in the endoplasmic reticulum will remove the first prefix, the pre. Then in the endoplasmic reticulum and in the Golgi apparatus, you do some post-translational modifications. For each signaling molecule, peptide signaling molecule, these modifications are different. For example, if we start with the insulin looks like this, the proinsulin. And if you do the post-translational modification, one thing is that you remove something that we call C-peptide. That means that the final insulin molecule is this, and you must join it by the disulfide bonds. So what you typically do inside is that you will typically cleave off some part of the molecule, you will produce the disulfide bonds inside, and do some other post-translational modifications in the endoplasmic reticulum and in the Golgi apparatus. And here comes the removing of the second prefix. That means that from the molecule of proinsulin, you produce the final insulin molecule. That means that from propeptide, you produce peptide, from prohormone, you produce hormone, and from proinsulin, you produce the insulin. And these post-translational modifications can be different for different molecules.
Where's this C-peptide come? Where does it happen? I read somewhere that it happens in a secretory granule.
Yes, that's the next step. That, of course, from the Golgi apparatus, you form the secretory granules, and that's the difference from steroid hormones and from thyroid hormones, that the peptide hormones are typically stored in the cell. So you do it subsequently in the process, in the Golgi apparatus, in the secretory granules, and different steps you do in the different parts. And as the final, you have in the secretory granules the mixture of insulin and C-peptide. And you produce, or do you secrete them from the cell in the equimolar quantity. That means 1 mole of this and 1 mole of this. This you do not use. You will measure it as the doctor to see what is the production of the insulin. But in the body, no physiological effect. And this is the final active molecule. For insulin, the story is simple. 1 protein or 1 proprotein, 1 signaling molecule. What is the second example is the example where 1 precursor, peptide, gives you different or more active molecules. So this molecule is called pro-opiomelanocortin, abbreviated as POMC, and familiar it's also called Big Mama. since it gives birth to 3 signaling molecules. The first is melanocyte-stimulating hormone. Second is the ACTH, that's the molecule that we mentioned today, that means adrenocorticotropic hormone, the hormone that commands to produce the cortisol. And the last is the beta endorphin, that means the endogenic opioid compound. 1 precursor, 3 different products. That means that in the next years, you will, for some crucial peptide signaling molecules, learn what is their precursor molecule. Whether the precursor molecule gives only the 1 possibility, or whether you can produce the alternative molecules. As for example, the glucagon, it's produced from proglucagon, but proglucagon can give you also, for example, molecules of glucagon-like peptides. Glucagon-like peptide 1 can be produced from it. So that means 1 precursor and alternative products. So do you understand this? POMC is propionmelanocortin, and it can be cleaved to these 3 molecules. That it means that it can give birth to more signaling molecules than 1. But all of them are peptides. Now we come to the secretory granules. Peptide hormones you typically store inside a cell. You store them, you can visualize them, so you can, for example, see insulin secretory granules. And these granules wait for the signal for the secretion to the blood. Once these proteins are inside the blood, they can be typically freely transported in the plasma, since they are hydrophilic. And since they are hydrophilic, they must have surface membrane receptors. And as you know, hormones typically do their action through 2 groups of membrane receptors. That means G protein coupled receptors, or by tyrosine kinases, or receptors associated with tyrosine kinases. You had a seminar about this, so we will not speak more about this, but typically for insulin, glucagon, growth hormone, if I mention only the peptide signaling molecules, you should know how they do the signaling inside the cell. If we now speak about the peptide signaling molecules, you very often in your books will read about 2 terms. There's the constitutive secretion and regulated secretion. Constitutive secretion of protein is typical, for example, for the plasma protein that you must continuously produce. That means that you do the constitutive secretion, and only small quantities of these proteins are actually stored, since you must, during the whole day, replace the lost proteins. For the peptide signaling molecules, you typically use the term regulated secretion. That means that these hormones are prepared in the store in the secretory vesicles, and after some signal, they are one moment at big quantities released to the bloodstream. And this signal can be, for example, for insulin, where you know it, is the increase of the blood glucose level. So if you have increase of blood glucose level, pancreatic beta cells will do the exocytosis of the secretory vesicles with insulin, and at one moment you will release quite big quantities. Or you can say in short time, you will release a lot of signaling molecules. Sometimes this regulation is done by some other signaling molecule. Yes? That means that you have the series of signaling molecules, and the signal for the secretion of the other is the presence of the previous signaling molecules. That's the thing that typically does the hypothalamus, pituitary gland, and the target organ, target endocrine gland. The last, how do we degrade it? Peptide signaling molecules. That's nothing new for you. You typically do the degradation inside the lysosomes, and the story is simple. You take the the receptor plus signaling molecule, that means insulin, for example, and insulin receptor. Then you do the endocytosis of this complex, and you, by the lysosome, degraded the signaling molecule, and the free receptors can return into the membrane. That means that here you typically do the degradation by the lysosomes. The enzymes are called proteases. Quite simple, similar for other extracellular proteins. Some small proteins you can find in the urine directly, but that's not the typical course of action how this is done. Yes, typically you degraded it in some tissues that have the predominant role in this. So any question?